All right, we are recording. So welcome everyone to the StaPH-B call. I think it's been a while since our last call, so I think we've probably picked up a number of new members. So for those of you who aren't familiar, this is the State Public Health Bioinformatics Work Group, and we have these monthly calls trying to focus on some of these topics that are relevant and consistent across the challenges for state public health laboratories in terms of integrating bioinformatics practices. Today we're going to talk to Jessica Chen from CDC's NARMS laboratory, where we're going to focus on antimicrobial resistance, determinant detection, and Jessica's going to talk about the workflow that she's helped develop and the sort of decisions they've made along the way and key variables to consider when you're integrating a similar technology or similar workflow. So from that, Jessica, I'm just going to give you the floor, and we'll have questions and time for discussion at the end. Take it away, Jess.
Awesome. Thank you for the invitation, Kevin. This is great to be able to speak to you guys about a topic that I really enjoy. So to give you an overview of what I'm going to go over today, I'll tell you a little bit about myself and what I do at CDC. I'm also going to tell you about the collaborative that I work for. It's called NARMS, and kind of our whole genome sequencing story and experience. I'll briefly go over the genomic basis of antimicrobial resistance, just so you kind of have appreciation for some of the decisions that went into developing our workflow, as well as some of the key considerations in building up our workflow. I'll go over our current workflow in NARMS in detail so you have a good idea of what software we're using and how we're going about that. And then I'll go a little bit into how we're using the data that we get out of this pipeline. So a little bit about me. My background is actually in food and animal science, specializing in microbial food safety. And then I started getting into kind of the bioinformatics space near the end of my PhD, during my postdoc, and during my time at CDC. So I've been at CDC for a little over 3 years. I came in late 2015 to work on antimicrobial resistance in enteric pathogens. And fun fact, I'm an enthusiastic bike commuter and cat fanatic, and those are my two cats, Sigma and Mew. So about NARMS, kind of who are we and what has our experience been with sequencing? So NARMS is the National Antimicrobial Resistance Monitoring System. We are a farm-to-fork surveillance system for antimicrobial resistance in enteric pathogens. This is made up of partners at 3 different US government agencies. The Centers for Disease Control and Prevention, where we're tasked with looking at AMR and enterics in human samples. The United States Department of Agriculture is tasked with looking at AMR in food animals, and the US Food and Drug Administration is tasked with looking at retail meats. So just to kind of give you an overview of where we were at before we went into whole genome sequencing, this is kind of like an outline of our phenotypic surveillance that we had in place and that we're actually still conducting to this day. So we conduct surveillance on non-typhoidal Salmonella, Shigella, E. coli 0157, and that consists of looking at 1 out of every 20 isolates that are submitted to state and local public health labs, and those are forwarded to CDC for AST testing. We also look at every typhoidal Salmonella and Vibrio non-cholera that is forwarded to us at CDC, as well as Campylobacter, and that scheme varies by state. Since Campylobacter is difficult to culture, not all states do it, and so therefore we only have certain participants sending us Campy. In terms of outbreak, our program looks at 3 representative isolates from multi-state outbreaks. And so a few years ago, we asked ourselves, and this is the collective NARMS this was actually before I joined, we asked ourselves, can we predict our AMR phenotype that we would normally get off of our traditional antimicrobial susceptibility testing? Can we predict that with sequence data? So our NARMS collaborative did a bunch of research in this area and ran some validations for Salmonella, Campy, and E. coli. And the conclusion that we came to was, yes, we can absolutely do this, and we can do it with high accuracy. So we've continued to be active in this area. Moving into the genomic basis of AR. So if you're not familiar with antimicrobial resistance, kind of to go into the biology just super briefly, bacteria can develop resistance in a number of ways. So the little pills in this schematic are antibiotics, and you can kind of see on the left, you can have enzymes in your bacteria that will hydrolyze or break down that antibiotic. You can have efflux pumps that pump it out of the cell. You can have mutations that occur that decrease the uptake to begin with into the cell. And then you can also have mutations occurring in kind of the target enzymes that antibiotics target within the cell to kind of evade the activity of the antibiotic itself. So how does that relate to the genomics? So here I have a schematic of a large chromosome and a plasmid that you may or may not have in your bacteria. And the colored boxes kind of represent resistance genes. So this is one way that bacteria can acquire resistance, is they pick up a resistance gene that encodes some kind of enzyme that confers resistance in some way. The other way that you can have resistance arise is through point mutations in chromosomally encoded housekeeping genes. So I've labeled these housekeeping genes in black, and that star represents a SNP mutation that may have occurred. So it's important to kind of look at two different rhythms that may occur in your cell. You would take kind of your favorite resistance gene database and look for these genes in your genome assembly, but you would also want to look for your favorite resistance mutations as well. And often this resistance, the resistance mutations are very frequently species specific, so you want to kind of take that into consideration. There are some good schemes out there for our bugs, but it may take some digging in the literature if you're targeting this towards some different pathogen. So briefly, not to like really get into detail on this, just to talk about AMR Gene Nomenclature, just to kind of warn you about what you're getting into if you go into this area. In the AMR world, some nomenclature is really well standardized and some is not at all. So for example, the beta-lactamase genes, these were well curated by the Leahy Institute, and now NCBI has taken this over, and this is super well defined. So you can be confident that a blah CMY2, it has a very specific definition. But then you move into something like the aminoglycosides, where there are two competing nomenclatures and nobody can agree on which one they want to use, and it's just frustrating. And some gene families are a huge mess because it really takes some individuals who really feel passionate about that gene group and kind of that nomenclature. So kind of very broadly to go over this, it's important to know there are what's called gene families. So the CMY2, the CTXM under the beta-lactamase genes on the left, that's the family component of that gene ID. And then the MCR1 and MCR2. So these are kind of distantly related gene groups. And then within them, you'll have a variant. So just kind of know that there are varying levels of discrimination within that gene name. So workflow considerations. So when we got started, which was also before my time, there were kind of three major players in terms of databases. There was Argonaut, CARD, and ResFinder. And we went with ResFinder, and I'm gonna walk you through the kind of decisions that we went through to kind of get to that decision. So we considered our application. What do we want to do with this? And it really came down to wanting to predict clinically relevant resistance profiles from sequence data. We also needed to consider our organisms of interest. So this, for our group, is predominantly Salmonella, Shigella, and E. coli. We also do a little bit of Campy and Vibrio, as I mentioned previously. There's also the level of curation and the ease of which you can modify the database. For us, our experience with ResFinder is the curators have been really responsive to feedback and quick to make changes. So if we identify a new gene and say to them, hey, we found a new gene, can you add it to your database? They'll do it right away. So kind of these things collectively together made us settle on this choice. And what it really comes down to is that the genes that we find out of the ResFinder database correlate really well in our organisms with our AST data. So this is really why we settled on this. And just kind of know going into this that no matter what database you use, some of the determinants in that database will not confer clinically relevant levels of resistance in your organism. So some notable examples in our bugs, in ResFinder, there's this AAC6'1AA gene. This is the terrible aminoglycoside nomenclature. It's present on a cryptic prophage that's in all Salmonella and doesn't actually confer resistance to aminoglycosides. It's inactive. Similarly, there's an MDFA gene that's present in all or most Shigella and E. coli, and it doesn't confer very high levels of resistance. It's a broad spectrum efflux pump and just doesn't really confer high level resistance and nothing that's ever going to cause treatment failure or register as resistant on our phenotypic panels. Point Finder, which is the point mutations scheme that we use for our bugs. There are several mutations that don't really cause change in phenotype, and I've listed one of these PAR-C mutations, and it's not associated with clinical resistance in our organisms. And the problem, I think, with this is that so much of the literature is based on these experiments where people would expose a bacteria to an antibiotic and look at the compensatory mutations, and not all of those are going to be the cause of the resistance that you observe. And it also includes some, the literature also includes some papers that are more observational studies where we found this mutation, but we found these other mutations too, and it was resistant, and we don't really know which one's causing it. So sometimes these mutations work their way into these schemes, and just know that it's going to take some trial and error to figure out what exactly is causing resistance in your organisms. One lesson that we learned fairly early on, kind of soon after I got to CDC, is that I think the low-level bleed-through that you observe with Illumina platforms really has to be mitigated if you're going to look at AMR gene detection. So in NARMS, the vast majority of the sequence data that we deal with is derived from Illumina higher MySeq, and the sample bleed-through is a well-known phenomenon. And you can see this Mitra et al. Paper from 2015 that goes over this super well. And it really has to do with kind of these sometimes reads from belonging to the wrong index get captured and binned in your sample in the image processing step. So this became a problem for us because we started noticing some genes present on very low coverage contigs that were not associated with their anticipated phenotype. So we verified this by PCR that these genes were truly absent in the sample. And we often found that these occur with genes that are typically on very high copy plasmids. So we then decided to establish this low coverage filter. So we set this threshold at 10% of the average genome coverage. So we'll omit any contigs from our assembly that kind of fall under that threshold. And I think this is generally a pretty good practice for a lot of different types of analysis, but it's especially crucial here. So I thought I'd mention it. So in terms of the threshold cutoffs that you set really depend on your application. So a lot of the tools that we're using are just very straightforward blast-based tools looking for genes in an assembly. So we decided to set these at 90% sequence identity for resistant determinant detection. And this allows us to capture everything with a relatively high similarity to genes in our reference database. And we also set the gene coverage cutoff at 50%. And people kind of look at me at this point and are like, you're crazy. Sometimes, and the rationale behind this is that sometimes resistance genes are present in multiple copies, and this happens quite a bit. and those don't assemble well and sometimes get split apart in your assembly. So having a lower coverage threshold allows us to capture more resistance genes that aren't assembling as well. And really what this boils down to for us is, in most cases, a family level gene identification, so not that specific variant, is sufficient to accurately produce resistance. So we can get away with these kind of looser thresholds, and we find that we're able to detect resistance very accurately. So kind of the perpetual question in this space is, do you want to do assembly-based or reads-based? Sorry, I've got a typo there. So you've got, your assembly-based tools like ResFinder, PointFinder, things that you assemble the genome and then run it through some kind of software that runs BLAST or some other method to search for your genes. So this, of course, requires you to produce the assembly first, so it's therefore a bit slower. But the upside of this is that you have an assembly to examine for the surrounding kind of gene context information. So you can kind of see our multiple genes being carried together in relatively close space in your genome. If you're lucky and able to assemble it well, you might be able to tell if it's on a plasmid or, in close proximity to a plasmid replicon. So it'll kind of give you an idea of kind of where those genes are localized. There are also kind of reads that don't require you to do a genome assembly, and tools in this area are Ariba and SRST2. These are much faster because you don't have to assemble first, and a bit more sensitive as you're not dependent on genome assembly. We ultimately went with an assembly-based method because you have to have, for us, with a lot of our downstream application, we have to have an assembly to kind of get a sense of kind of how these resistance genes are localized, how they're moving about in a population. So having that assembly is really great for us. So we decided to go down this route. And ultimately, with our fairly relaxed cutoffs, we don't see a lot of things that are being missed or kind of assembled out of our genome. So kind of digging into our current workflow in use. So we get the data off of our sequencer and we do some QC steps. We run it through CG pipeline to compute standard read metrics like Q-score and sequencing coverage. And we typically we always confirm sequence ID with Kraken, and then with Salmonella, it would be like Seq02 to verify that it is what the organism we think it is and the serotype that we think it is. So I'm not going to go over Kraken and Seq0 today, but I'll talk a little bit about kind of why we use CG pipeline and what for. So I use this with the fast option, and this computes the metrics on a subsample of reads, so it's quite quick. And we feed in the genome size of our organism of interest. And this is where we calculate the coverage, so we have a more informed information on what our coverage cutoff threshold will be when we go to assemble. So next we assemble with Shovel, and Shovel is a kind of quicker version of Spades. We think it's really great and have had a great experience with it. The options that we use are below, and you can see we set the genome size, the minimum coverage, so contigs below that threshold are omitted, and as I mentioned previously, we omit things that are 10% of the average genome coverage. We use the trim option with Trim-O-Matic and the name format option to make the contig headers pretty. So to do the actual detection, we've been using a program called StaR-AMR. This is a rewrite of ResFinder and PointFinder that was developed by some of our collaborators at the Public Health Agency of Canada. StaR-AMR has this search function, which we use with the -- PointFinder organism, and this turns on the resistance mutation detection. And then you just specify the scheme that you're aiming to run. So in this case, it's Salmonella. You set your % identity threshold and your gene length coverage cutoff. And by default, the really nice thing about StaR-AMR is that they will exclude genes listed in this genes to exclude.tsv that I mentioned at the bottom. So by default, this comes with that AAC6'1AA gene that's present in all Salmonella but not functional. But you can also pass a custom list using the -- exclude genes file option, and I think that's a really handy feature of this software. And so this presentation isn't about plasmids, but I think that I should mention it because it's something that we do as part of our routine characterization. We have a custom plasmid database that's developed off of Plasmid Finder. We've updated this to include, one of my colleagues has updated this to include more current plasmid nomenclature than what's reflected in Plasmid Finder, and it includes a couple of custom targets that we've that kind of encompass a few plasmids of interest that we see routinely. And we've submitted these changes to Plasmid Finder, but they've yet to do anything on this. So hopefully it's something they'll do. If not, I think this is something we'll try to make public. The options, you just set similar options to what's in StaR-AMR, just the min ID and the min cov. And we use a little bit higher gene coverage threshold for these plasmid genes because they are quite small. So having the coverage being lower than 60% would give us a lot of noise. So let's briefly talk about Campylobacter since that was the Salmonella scheme, just because there are some interesting caveats and adjustments that we had to make to modify our workflow to make this work for Campy. So it's largely the same workflow as Salmonella, but one big difference. Mutations in the 23S rRNA gene in Campy are very important for this resistance in this organism. These mutations confer resistance to macrolides, which is an important treatment option for severe Campy infections. So you can see in the schematic on the right, I've got a genome, and these 3 boxes are your 3 copies of the 23S rRNA gene. But a SNP mutation in only one of these copies can produce the phenotype of interest. So you can see right away that there's probably an issue with using an assembly-based approach on this. It's just not reliable at all. This region doesn't assemble well, and even if it does, if only one of your copies is mutated, you may not even see that mutation in your assembly. So we had to go back to the drawing board on this. So we still use StaR-AMR to detect the resistance genes, but also some of the other resistance mutations in single copy genes. But we feed in this Campy genes to exclude TSV, and that includes the 23S target that we no longer want to call from the assembly. And then we created a custom database in Ariba that includes a reference the same reference 23S gene that's in the point finder schematic. And we run this on raw reads and are able to pull out our mutation of interest with high accuracy with this. So very much similar to our approach with Salmonella, but a few small tweaks, and this change should detect these multi-copy genes with a reads-based approach. So what do we do with this information? We've got a list of the determinants out of StaR-AMR and sometimes Ariba. Where do we go from here? So with all of our experience and kind of the information that we've gained over the years, we can start identifying a predicted resistance pattern based on the wealth of data that NARMS has. So on the left, you see the genes that you might detect out of one of these softwares. And then we can start assigning a phenotype based on the presence of that gene. So we just have a lookup table that we use to kind of predict the resistance from these. And this is implemented in our IT team's access database. So it's not something that we do in-house. We just upload our gene calls to our database, and we get this predicted resistance field populated, which is really great. And another thing to mention is that the StaR-AMR software will also assign predicted resistance values, and this is based on the NARMS drug key that we've developed. So we shared this information with them, and they've made it public on their GitHub. So how we use this data. So you may have seen some of the outbreak notifications that, and the investigation notices that CDC now puts out. This is one from last year that involves multi-drug resistant Salmonella in raw chicken. And you can see here that we have put some of our information in here, this WGS analysis, predicted resistance to these antibiotics is a huge long list. This is a really nasty bug, but we aim to make that information as public as possible. And then we have down here a link through to advice to clinicians so that it's in a kind of more easily digestible format. You know, use these drugs, don't use these drugs. So that's one key place where our data gets used. We then publish a summary of our surveillance each year in the NARMS Human Isolates Report, and then we work with our partners at FDA and USDA to publish this information in an integrated report format. But we also conduct research, and kind of the key areas that we're interested in are exploring the emergence of rare resistance determinants. So if we see a gene that we've not seen in the US before, we'll often try to have a look at kind of the context surrounding that. Maybe we'll do some long-read sequencing to kind of close the plasmid or whatever mobile element is responsible for bringing that gene in, trying to find some exposure information from the patient. So we have an epi team that we work really closely with to kind of get at some of these questions. We're interested in understanding the spread of multi-drug resistant strains of enteric pathogens. So to the right, you can see a study that we published last year that looked at kind of a molecular clock and a temporal phylogeny of this multi-drug resistant strain of Salmonella infantis, and we were able to kind of link our data, which is in black, to some sequences from South America, which are in blue, and can kind of see that it appears that the strain originated in South America before it appeared in the US. So it's that kind of getting at those stories and finding out how things become introduced in humans and into the food supply is really of interest to the NARMS group. We also do work in kind of looking at new resistance determinants by closely examining sequences from isolates with genotype-phenotype disconnects. So since we're still doing phenotypic testing, sometimes we'll see a phenotype but don't see a responsible gene, and that's kind of a signal to us that maybe we need to take a closer look at this strain. So hopefully at this point I've convinced you that WGS is highly accurate and a really great way to look at AMR. But our lab, in the position that it's in, is actually not going to do away with phenotypic testing. I will say that with the PulseNet transition to WGS, we're now getting a lot more resistance data. So remember that we only get 1 in 20 of Salmonella submitted to state public health labs. And now with kind of the data that PulseNet's analyzing, we're seeing a lot more Salmonella sequencing and getting AMR information that way. So this allows us to identify really rare occurrences, so super rare genes that we don't see very frequently, and also gives us some increased statistical power to look at changing trends. However, from kind of the NARMS standpoint, it's important for us to maintain phenotypic testing as kind of a sentinel surveillance for new and emerging trends. Right now, a lot of the detection is database dependent, and we're going to use that we're going to use our phenotypic data to continue to examine instances where a phenotype exists without a genotype. And actually, an example of this is last year, my colleague Caitlin Tagg identified a novel trimethoprim resistance gene, DFRA34, and we published that, and it's out there, and that gene is in ResFinder. So we are going to, so we found this because we saw from our phenotypic testing, hey, this is trimethoprim resistant, but there's no DFR genes. So we took a closer look and were able to find this one. And also a notable example is mobile colistin resistance, or MCR1. That was identified because a stable phenotype was detected first. And that's when researchers really dug into the sequence and was able to find what was responsible. So I think that these things go hand in hand, and we're able to use both data really well together. So I just want to say thanks to all the participating public health labs, both submit sequences to PulseNet, but also submit isolates to our lab. We would not exist without you, so thanks. And then the other members of our NARMS collaborative, FDA, our partners at FDA and USDA, they do a lot of work in this realm too. And then shout out to my colleagues in both the NARMS teams at CDC. We have a NARMS EPI and a NARMS lab team, and this work would not be possible without them. So. And with that, I'd like to open up to questions or discussions. And if you think of anything kind of afterwards, my email is lly3@cdc.gov. So feel free. I'm always happy to geek out about resistance. So thanks.
Awesome. Thank you, Jessica. And then, yeah, definitely have a couple questions off the bat. So I just wanted to start it off here. So you were talking about a high correlation between the genotypes and phenotypes that you guys had already seen in the thousands of isolates that you've analyzed. Can you talk about what those correlation numbers look like? And also, what does it mean to correlate? Is it once intermediate or like decreased susceptibility is identified, or is it clinical resistant levels, and go on from there?
Yeah, that's a really good question. So we look at, we aim to compare mostly just kind of the resistant class to presence absence data, kind of that predicted resistance. One notable exception is we'll look at intermediate resistance to ciprofloxacin. There's genes that produce just intermediate level to CYP, and that's very, very well defined. It doesn't usually push it to full resistance. So we have that coded in our database as like CYP intermediate or resistant. In terms of the correlation, it's well over 98%. For our 2016 data, we actually went through and resequenced a number of isolates that were giving us trouble and did re-AST and came down to only like 6 disconnects out of like 3,500 isolates. And these were like single drug disconnects in a strain. So it's really, really accurate. And when we see a disconnect, that's often a sign that there's something going on. So if we see a disconnect, we'll retest that for AST to make sure that that phenotype is stable, and then we'll start digging into the genome and consider closing it and looking at it in more detail.
Okay, and when you have that 98%, is most of these the discrepancies then are like false negatives in terms of not finding a gene that would confer that resistance? Or are you also seeing the opposite, where you're seeing a lot of these resistance genes, like you mentioned, some of those aminoglycoside genes in Salmonella? Are you seeing
Yeah.
that as well?
So we have that notable example kind of omitted from our schematic, the notable aminoglycoside, just because we know it's not an active gene. Sometimes, yes, we'll see it in both directions. Sometimes we'll see a gene that's maybe interrupted by a transposon, but that's actually, it's pretty rare. We don't see it happen that much because usually that gene is there for a reason. It's producing resistance to an antibiotic. It's giving it some kind of selective advantage in that environment. So yeah, so sometimes they are interrupted, and sometimes, yes, we see something where we have a phenotype but no gene. Another thing that we have an issue with is sometimes the plasmids aren't super stable. So just in the time between we do AST testing and sequencing, you might lose a plasmid, which we can then confirm by doing re-AST on the sample. You can usually see that it's lost, or and yeah, that's.
Okay, definitely. Thank you.
Hey, Jessica, this is Kelly from Utah. Question: if I wanted to get my hands on that plasmid database, could I get that?
Very likely. So I'll ask my colleague, but I'm sure that won't be a problem.
That would be awesome. Have you guys thought about or have tried using plasmid spades to assemble plasmids away from chromosomal? And if so, has that worked for you at all?
We have used that, and I'm glad you brought it up. There are a lot of interesting tools in the space, and we're really interested in plasmid assembly from short read data. The issue with plasmid spades, sometimes we will get a great assembly, but there has to be kind of a coverage difference between the plasmid and the chromosome. So if you've got a plasmid that's at that has an extremely high copy number that's in really high abundance in the cell, the plasmid-Spades algorithm can sort that out really well. One of the interesting things that we've been playing around with, though, is PlasFlow. So this is a machine learning plasmid software, and so it will kind of predict what contigs based on a plasmid database are likely belonging to your plasmid, and then it will kind of infer what's chromosomal. And then we take it a step further and map reads to the chromosome and put all the unmapped reads into another file and try to reassemble using unicycler. And sometimes that gets us really good results, and sometimes it's just like, you know, we have to just do long read sequencing on it.
Yeah. Okay. I have a bunch more questions. I can keep going unless somebody else.
No. I got the next one.
Okay, there you go.
Hey Jessica, this is Sean from Minnesota. I mean, since you sort of like agreed to share the plasmid database with Callie, why don't we just, you know, put one step further and just put it on the StaPH-B GitHub? That will benefit all of us.
Yeah.
And my question, have you tried the long reading sequence? I'm sure Paris will be excited.
Sorry, I can't hear you super well. Can you speak up a little?
Yes. So I'm curious, have you guys tried the MinION, you know, long reading sequence to pick up the plasmid?
So I think I heard you ask, are we trying MinION sequencing?
With the plasmids, yes.
With plasmids. So we do have a little bit of AMD funding in collaboration with some of the folks in our reference lab to look at this. So we will be trying to use MinION to get some assemblies of plasmids. So yes, we're headed in that direction. Right now we just send isolates to our core facility and they do PacBio. So that's typically what we do, but if we can get MinION to work, that's awesome too. And yes, to respond to your comment about putting it on GitHub, yeah, I will talk to my colleague that developed the database, and I think that's definitely a great option for us. But I think we're more than happy to share. I don't want to speak too much for her, but I think that it shouldn't be a problem. We've shared it with many other collaborators, so this shouldn't be an issue.
Thank you.
Go ahead, Sean, sorry. Okay, so
No, actually.
just to tag in onto Sean's comment there about the MinION and plasmid sequencing and closing, I'm wondering how epidemiologists would use that information as well, like knowing whether it's plasmid mediated or chromosomally mediated. Are these things that you also include in those outbreak reports?
We don't include that information in our outbreak reports, but it's something that our epis are very, very interested in, kind of knowing if something is mobilizable or not is of special importance. So definitely having that information, I think, is really helpful to us in terms of understanding how things move about.
Yeah, okay, thanks.
Hi, this is Logan from Colorado. Great talk, by the way. Really interesting. I love AMR stuff. I think it's fascinating. So, but I was wondering, do you guys have that list of the genes that you exclude, and can you make that public as well, and maybe put that on the GitHub as well? Just
Absolutely.
because I keep getting those kind of genes as well, and it's nice to have something to kind of like sort through and be like, okay, this is probably not, you know, as important as maybe some of the other genes.
Sure, absolutely. We can make that available.
All right, thank you so much.
And just so you're aware, Jessica, we have like a StaPH-B GitHub page, so I can be the sort of liaison in that respect. And I think you've actually sent me a lot of those details
Okay.
about modulations of the databases that you've made. So I'll be sure to make that public to the group.
Yeah, I can help facilitate that as well.
Awesome.
That'd be great.
Appreciate it, guys.
Yeah.
So I have a question. So do you ever collaborate with the hospital-acquired infection group there to look at resistance in non-enteric pathogens?
Yeah, so we talk extensively with them because they're interested in very similar things. Kind of a particular area of interest is metagenomics. That's kind of where we overlap quite a bit because we're, definitely thinking about the world without culture and what that will look like and what that will do for our analysis. So we are definitely in contact with them about kind of ways that we can prepare for that and think about how to look at things metagenomically. But also sometimes there are things that overlap. We have some initiatives to look at MCR1 together. So whenever we get an MCR1 case, positive case, we share that with the hospital acquired group and vice versa. So there, yeah, there's definitely a lot of dialogue, and we definitely work very closely with them.
And then also, what about like environmental stuff? Do you guys work with Genome Tracker and those folks to get a picture of what's happening out in the environment?
Yeah, so we collaborate with FDA. So most of our partners are at the Center for Veterinary Medicine, but they also work very closely and analyze a lot of data from Genome Tracker and CIFSAN as well. Definitely we have very active collaborations with FDA and the Genome Tracker initiative.
Cool.
In terms of other environmental stuff, we are collaborating with the waterborne group at CDC to kind of look at kind of things in the natural environment. We've got a pilot project in collaboration with waterborne looking at resistance in the Chattahoochee River in Georgia. We have been having discussions with folks at EPA to look at kind of rivers and streams in the US as well. So kind of definitely have a lot of communication with other partners outside of those that are formally part of our NARMS group.
Yeah, that's going to be an interesting one, looking at environmental isolates too, and sort of how they would fit in these known databases, because having some experience with environmental isolates, you just see some variants that have just otherwise not been characterized.
Definitely.
And also,
Definitely, yeah. Go ahead.
I would say there's questions on the chat. I don't know if you can see those, but we have something from both Sean Wang and then Wyoming Public Health Laboratory there.
Yeah, so we don't collaborate directly with ARLN, but we do get information from them. So sometimes things will accidentally get submitted to ARLN that are supposed to come to us, and vice versa. So we talk with them quite a bit. Really have a super active collaboration with them right now, and I don't believe we have any collaboration with ARX at the moment either. And then in terms of transcriptomics, that's a great question. We haven't had anything quite yet that we've wanted to take a look at, but I could see how it would be useful in some of these cases where we kind of see something that is present without a gene that we know of, but and we have like a phenotype but no gene present. It might be interesting to kind of see what's going on at the transcriptional level, but we haven't yet needed to really dive into that too far yet.
I have more questions if
Sure.
we have time. Okay, so I had a question about the resistant determinant cutoffs. So when you use that like 50% coverage of a gene, how often do you go in and see that that was like a sequencing error or like an assembly error versus just like a random, you know, like 50% hit that doesn't mean anything?
Yeah, by and large, it's something where it's just an assembly issue that there either will, like if we long read sequence it, there'll be multiple copies present and it's just not being assembled super well, or it's just period not being assembled well. It's, I think, pretty rare that we'll see only like 60% of the gene, 70% of the gene. Sometimes we'll see stuff where, you know, a transposon has inserted, so you'll get two different, you know, pieces in the genome and it has to do with that insertion.
And then do you guys have like a way to look for transposons specifically and that kind of stuff? Or are you monitoring for that when you find those kind of like?
So my colleagues are using, this is a little bit out of my area expertise, but I can definitely speak to it. My colleagues are using what's called Galileo AMR. It's not a free software, but one of my colleagues, her major professor, was instrumental in developing it, so she has like free access for life. So that does really high quality annotation of resistance regions. But also they use IS Finder as well to look at for insertion sequences. But yeah, Galileo AMR is definitely their big thing.
Thank you.
So Jessica, I had a question on the lookup table too that you guys
Sure.
had. I noticed that there was some aminoglycosides, or at least even all of them, where the phenotype prediction is relayed to a very specific drug. So it's not like the drug class. So
Hmm.
in that case, are you seeing high correlation? I think generally I see like a gene associated with a class, like aminoglycoside, for example, and that's kind
Yeah.
of where the correlation gets drawn. But you're seeing it all the way up to the specific drug type.
So, I'll take a step back and kind of explain how we develop the drug key. So we basically just take everything that's in ResFinder and PointFinder and develop a prediction for that. And the thing about enterics is that we see a lot of the same genes over and over again, and some of the things in the ResFinder database we're not ever going to see really. So our prediction is highly accurate for kind of the drugs that we test for and the things that we routinely see, but it's kind of hard to say what the accuracy would be in a different organism or for some of these things that we've really never really seen before. We actively work to kind of keep up on this drug key. So if we see something come through that's been sequenced like at a state lab that has a gene we've never seen before, we'll reach out and be like, hey, can you guys send us that? We'd love to test it.
Yeah.
Because then we have that information. So kind of the first pass information are like, here are the genes that we know confer this defined phenotype. And then then it's digging through literature and kind of like sometimes you'll strike gold and find something that is in your target pathogen in a super well conducted study that looks at your drugs of interest, and we are able to say pretty definitively like this is going to cause this resistance. But other times it's just like, well, we think that it would confer this resistance, but we're really not sure. So that's kind of the basis of it. It's imperfect, but it's something that we're constantly working to improve. So if you happen to see anything of interest that, seems super rare, we'd love to hear about it.
Okay, thank you.
All right, I have one more.
Okay.
So you talked about you only had to have one point mutation in 23S in Campylobacter to convey resistance, right?
Yes.
How do you get around the idea that at least all 23S's should be all the same, because homologous recombination has been basically removing any point mutation that you should get there, right? So do you
Right.
always see it in one copy, or do you always see it in all three copies?
So
And if so, how do you determine which copy it's actually in?
yeah, so that's a good question. Usually we see, like, when I look at the read mapping data, 100% of the reads are showing this mutation. That's like the vast majority of cases. We've only had a few occurrences where, you'll see like 30% of the reads. And we assume that that's one copy because it's a third, but we know that that coverage is kind of imperfect. We usually don't really go in there to nail down what specific copy it is. It doesn't really matter for our prediction. Just kind of knowing that any copy may have this mutation is beneficial to us.
Okay, awesome.
I have something. It's not really a question, more
No
just
worries.
something I learned today.
Okay.
So some of my colleagues here in EDLB were just at SFAF, the conference, and they'd spoken with Jonathan Jacobs, and so he works at QIAGEN developing CLC and stuff, and he told them that at the ASM meeting upcoming in June, they're planning on making an announcement for a kind of a gold standard AMR database.
Mm-hmm.
And so apparently he's been going around to all the different database people, ResFinder card, like you name it, he's talked to them and worked on like licensing the databases. And basically they're trying to curate one, unified nomenclature, one database. So just be on the lookout if anybody's at ASM for that. And then I think they're planning on releasing it, I think they said late, either this fall or early spring. as sort of like an add-on to CLC Workbench, like a free add-on if you've got the software. But I don't know if anybody had heard more about that or has talked to Jonathan or anything.
Yeah, I've seen him tweeting about it quite a bit.
Yeah, he tweets a lot.
So yeah, it sounds like that's coming out this summer, and I'm really interested to see what's there. And actually, just earlier this week, I got asked, the folks at NCBI reached out to ask us to look at their latest version of AMR Finder as well. And I think that this one has point mutations built in, which is an improvement over their last version. They also, another kind of nice thing that NCBI has is they have sanitizer resistance built in. So if you're interested in those things, that can be really great as well. We don't look at that quite so often because we're really bottom line interested in sanitizer or in antibiotic resistance. But in kind of a more research space, understanding what is kind of carried alongside resistance determinants, that's something we may start to dig into a little bit more.
Yes, I'm actually really glad you brought that. When you say sanitizer resistance, like the gene that pops up in my head is like Quacky Delta. And yeah, so especially as you get more involved in plasmids and investigating those, that does seem like something to me that would be really valuable to know if it's there, because it's such a strong indicator of the presence of like a class 1 integron.
Mm-hmm.
Yeah, I don't know if you could speak about that in terms of, well, that's something we saw when we were trying to identify the proper database. Like if you look at NCBI, they include these things, whereas in your lab or ResFinder or other databases, they exclude these things.
Yeah, that's something that we definitely see when we're looking at, when my colleagues are looking at kind of that fine-tune resistance annotation using Galileo AMR. They will often see that as part of a class 1 integron, absolutely. Yeah, I think it's a really, really interesting area. It can give you kind of an idea of why that plasmid might be maintained in that organism, because you think about some of these large plasmids, it can be a bit of a metabolic burden. So there has to be some benefit to having these plasmids and keeping them around. So it's also really interesting to us to know what's co-localized alongside our AMR genes. So yes, one of the latest versions of Proca definitely has IS Finder built in. So that's something that we've been looking at as well. That's a good comment, Kelly. Any other questions?
Yeah, I hate IS elements. I literally cannot stand those little buggers.
Yeah, they're really challenging, and I think that IS Finder is kind of tricky to use and interpret. So that's yeah.
Yeah, it's great if you want to submit a genome to them. They'll send back a really nicely well annotated version of your genome with IS elements. They'll even go on and find direct and indirect repeats and tandem repeats and all of that stuff. But it's beyond the scope of what any of us do in public health. But yeah, it's
yeah. I have
IS elements are a bear.
Yeah, I have some colleagues that do a lot of work, and looking for those direct repeats and looking for those IS elements, and they refer to it as an artisanal process because we just don't have a super great way of doing that in a high throughput setting.
All right, any more?
Yeah, I spent a year and a half annotating a genome just to find IS element tandem repeats. So. Yeah.
I had one more question, and it might be a little bit like not something that's that relevant, but you were talking about with the point mutations. Sometimes they'll come in like groups. You'll have like, oh, this bug has multiple point mutations, and then you're not sure which one is actually causing it. So then in the database, do those then just kind of get lumped as like one genotype that would be correlated to a phenotype, or do each one of those things kind of get their own specific entry, and then those will pop up individually each time afterwards?
So they'll each get their own specific entry. So we'll get a list out of, we see an amino acid change at these positions. And, for the vast majority of our bugs, there are like 3 point mutations that cause the majority of the resistance. So it's not, we see some one-offs, kind of like something that we might see once a year, and this super rare mutation will pop up, and we now know that causes resistance because we've seen it and we've examined it more closely. But yeah, like really, it's like there's like a few key areas, especially for quinolone resistance, that just are responsible for the vast majority of resistance, at least in Salmonella, Shigella, E. coli.
Jessica, I have one more question to wrap up the hour here. I'm wondering from the NARMS perspective, after doing this for as long as you guys have done this and confirming like the strength of this kind of technology, what sort of either public health change or antibiotic stewardship change do you see happening as a result of these kinds of programs?
Yeah, that's a really great question. So I kind of highlighted an example of kind of how we've used this data to kind of promote changes. For instance, the Salmonella infantis, the multi-drug resistant bug that we saw last year, we started seeing it kind of pop up in poultry samples. And so we had a meeting with the Chicken Council and said, hey, we're seeing this problem. We're seeing this uptick. Like, we're confirming this. It's phenotypically there as well. It's not just genotype. Like, let's talk about this. So we've actually started, and we've had similar discussions about a different strain of Salmonella with the Turkey Council. So I think that kind of talking with those industry groups has been, I think, is a really great way. Kind of give them an early insight into kind of what we're seeing, the changes that we're seeing, the potential problems that are brewing, and kind of giving them an idea of what's going on, and allowing them to kind of adopt changes has been beneficial, and I think will be beneficial going forward.
Yeah, that's fantastic. I didn't realize you guys were having those kinds of meetings already. So it's great to hear that both those count. I didn't realize there was a poultry council.
Yeah, sometimes you don't realize these things are out there until like an outbreak happens or something, and then you kind of start like looking like who represents all the chicken farmers and producers, and then you figure out that these organizations are out there.
That's great, and it's great to hear that there's a consortium of people out there, and that they're also receptive to understanding the really cutting edge science of what we're detecting, and open to making changes. So we're at the top of the hour. I did want to say thank you again, Jessica. That was a really great talk, good insight, very stimulating questions and conversations throughout. We're going. Oh yeah, and Sean recommended that book we were talking about at the beginning of the hour, the Big Chicken book. But yeah, so all those questions and databases that Jessica's lab has been working on, I'll make sure to speak with Jessica and Curtis also to make sure we can get those on the GitHub pages. If you guys have any other questions, again, there's Jessica's email, and that's LLY3?
Yes. Yep.
Okay.
Lily without the I, 3.
There you go. All right. So thank you guys for another great StaPH-B talk. I'll go ahead and put this on the Collaborate page when I have the chance to. So have a great day. Thanks again, Jessica. And
Perfect. Thanks for the invite.
Of course.
Thanks everybody for tuning in.